So to summarize this portion of the talk, very different from what we saw in renal cancer, the real collaboration in this case seems to be between HIF1 and, in this case, Myc. Myc amplified tumors preferentially express HIF1 and not HIF2. Myc overexpression, in this case, therefore, can maintain neuroblastoma cell growth and hypoxia. And HIF1 and Myc can actually collaborate to promote the expression of a number of glycolytic enzymes or things that modulate mitochondrial activity, making this another class of tumor that might be ripe for therapeutic intervention by inhibitors of lactate dehydrogenase A activity. And so what we would argue is that in a tumor, which as always would have very steep oxygen gradients, in normoxic conditions where oxygen and perhaps other nutrients are available, the high levels of Myc activity promote both proliferation, enhanced glycolysis, and tumor progression. But this extends to the hypoxic subdomains because the Myc amplification can override the inhibitory effects of HIF1, and this allows the tumor to sustain proliferation, and then the two factors, oops, I think my thing died, so I'll try this one. And so these two factors then can collaborate to enhance the expression of a number of glycolytic enzymes. But of course, we wanted to turn our attention to the glutamine pathway for the reasons you've heard from multiple speakers, assuming that these cells would also be very dependent on glutamine uptake, glucose and glutamine being so important for central carbon and nitrogen metabolism in cells in a highly proliferative state. So as we've already heard, Myc or Myc, or I should say Myc expressing cells are addicted to glutamine. If it's withdrawn, they can undergo apoptosis. Myc can impact glutamine metabolism at multiple points. And so we wanted to ask two basic questions. It's very clear that these cells become very dependent on glutamine, but what are the mechanisms whereby glutamine deprivation induces Myc or Myc overexpressing cells, and can we actually implement this in targeted therapeutic approaches? And the idea was to come up with a way of providing synthetic lethality with Myc, that is, in the cells that are exhibiting deregulated expression of this oncogene, these cells would be selectively targeted by very specific drugs. And we think we found something that may be useful in the clinics, and so I'm going to use my last few minutes to describe that work. So now, once again, returning to the large bank of neuroblastoma primary patient tumors, we showed that they did not seem, when Myc was amplified, express terribly high levels of glutaminase 1. Oh, thank you very much. That's better. But they did express very significantly higher levels of at least one of the high-affinity glutamine transporters, as well as somewhat higher levels of glutamate oxaloacetate, too. Just as has been shown by David Weiss and others in Myc overexpressing cells, when glutamine is withdrawn, these cells tend to die. So Kelly is a cell line derived from Myc amplified primary tumors, and this extended to other examples of cells created from tumors that exceedingly high levels of the Myc oncoprotein. This is dependent on apoptosis because it can be inhibited by a broad-spectrum caspase inhibitor, that being ZVAD. And not too surprisingly, it is dependent on one of the pro-apoptotic proteins. We showed by conformation-specific antibodies that BAX in particular is significantly activated upon glutamine deprivation. BAX is to some extent, and much to my surprise, this was really BAX-driven, but somewhat neutral for BAX. So siRNAs to BAX totally reversed the cell death response upon glutamine deprivation, but BAX seemed to be somewhat neutral. Unlike BAX can be found in cytoplasmic reservoirs in association with proteins like Ku70, and this association can be inhibited by proteins like CBP, so now BAX oligomerizes and induces cell death at the mitochondrion. But in contrast to control experiments, we saw no role for Ku70 acetylation in this process. So we turned our attention to upstream activators such as BIM, BID, and PUMA, and showed that in glutamine-deprived Myc transformed cells, both PUMA and NOXA, were upregulated at the RNA level, and PUMA in particular at the protein level. And PUMA inhibition actually can ameliorate some of the apoptosis that ensues upon glutamine starvation of these cells. Now, PUMA and NOXA are well-characterized p53 targets, and neuroblastoma frequently has at least a somewhat intact p53 pathway, but once again, we found that p53 was not involved in PUMA engagement upon glutamine starvation. And we learned that the PUMA locus actually harbors a consensus for a very important regulatory site that's engaged upon amino acid deprivation. This activates a factor called ATF4, and it responds to this nutrient stress by upregulating amino acid transporters and other enzymes like asparagine synthase. And so I will, in the interest of time, gloss over this slide, but we've been able to show that ATF4 is activating PUMA. We can chip ATF4 at the PUMA locus in glutamine-starved cells along with RNA polymerase II. So we argue, and this would be consistent with what you've heard before, that the reason that the cells are dying is because of TCA cycle collapse, and we can reverse the cell death by now providing the glutamine-starved cells with membrane-permeable versions of oxaloacetate and alpha-ketoglutarate. So this is intriguing because it appears that under these circumstances, glutamine has been converted to an essential amino acid, engaging aspects of the ER-resident integrated stress response, and we don't understand how ATF4 mRNA levels are going up, but it would be very logical to assume that its rate of translation is being enhanced if cells are experiencing amino acid deprivation. And in fact, this is the case. We've been able to show, and you can see how ATF protein levels go up significantly as glutamine starvation ensues. We've used MEFs that lack one of the upstream kinases that regulate selective ATF4 translation, that being GCN2, or MEFs that lack PERK, and this definitely tracks with GCN2 kinase activity. And we've also been able to modulate PERK and GCN2 back in the neuroblastoma cells themselves. And again, it's consistent with enhanced translation due to GCN2 kinase activity. So I'm just about out of time. People might be surprised by this because ATF4, upon nutrient deprivation, usually results in a survival response because these downstream targets would promote amino acid uptake and novel amino acid synthesis. But we suggest that in a very situation where you have an extreme dependence on glutamine uptake and high rates of proliferation, that ATF4 is instead converted to something that promotes death. The enzymes that promote glutamine metabolism include glutamate dehydrogenase and glutamate oxaloacetate transaminase, and there are small molecule inhibitors of these two enzymes. And in the last slide, I'm simply showing that either of them can restrain subcutaneous tumor growth, and especially when combined with another molecule called fenretinine, which specifically activates ATF4 activity because of an imbalance in ceramide metabolism. And so fenretinide has been suggested to be something that could be useful in the clinics, but it's met with, I would say, rather disappointing results. But perhaps ATF4 agonists like fenretinide could now be combined with components, with inhibitors of components of the glutamine metabolism pathway to come up with a new way to treat patients that exhibit deregulated Myc or Myc. And so I will skip over this because I've already made all of those points, but just to remind everyone in the final slide, we all appreciate that Myc can either promote proliferation or apoptosis depending on the signal, and in situations where glutamine is present, the Myc pathway can harness glutamine to its advantage to promote cell proliferation, but if glutamine is absent, this can result in cell death. And then coming full circle to the availability of oxygen and nutrients in tumors, we believe by now treating these tumors with things that disrupt glutamine metabolism and promote ATF4 activity, we could extend the cell death into the regions of the tumor that have reasonable levels of oxygen and nutrients. So we hope that this will actually have some usefulness, or the ideas of this will have some usefulness in the clinic at some time. And I'll just acknowledge members of the laboratory that did the work. John Gordon really got us started on looking at crosstalk between HIF and Myc and renal cancer, and pretty much everything I described today is the work of one postdoctoral fellow, Guoling Qing. So thank you very much for the invitation to speak and your attendance, and at the break, like Craig, I'm always open to any and all suggestions or comments. Thank you very much.